been at Portland State now since 2003. His lab has been, I would say, extremely well funded by the NIH and NSF. And he holds editorial positions on multiple different journals, and is currently the chair of the Comparative Physiology and Biochemistry Division at SICB, which is what we have been collaborating on recently. And we're very happy to have him here to talk to us about extreme physiology.
All right. Thank you. Thanks, Jamie. Thanks for coming and for inviting me. And I got to tell you, it was weird to walk into Cordley Hall because it looks way different than it did 30 years ago, for sure. It's a beautiful building. All right. So today I'm going to talk with you about my work that's focused on developmental ecology. So looking at how embryos actually interact with their environment as they're developing. And I got interested in this because I took developmental biology, and it was super interesting to think about how you could go from, a single cell to this multicellular organism faithfully, generation after generation. And we were looking at all of these model organisms that were grown in the lab under controlled conditions. And I thought to myself, no organism actually develops under controlled conditions. So how do you then layer upon that whole really complex program of going from a single cell to a multicellular organism? How do you layer on top of that the effects of the environment or ecological interactions and still get a faithful outcome? And I got interested in these annual killifish I'll talk with you about today because they live in ephemeral ponds. So this is an actual killifish pond in Uruguay, and they're essentially flooded fields. They're very shallow. They go through extreme temperature and actually almost all environmental parameter changes every day. It rivals the intertidal and other places like that. So they might be pretty much anoxic at night from all of the algal growth and things that are in the pond, and super saturated with oxygen during the day for the same reason. The pH shifts every day from like 5 to 10 on a daily basis. Temperatures go from the low to mid 20s to the high 30s. Some of them were up to 42, 45 degrees Celsius in the afternoon. And the sediments, the place where the embryos live, are anoxic. These sediment PEs are negative or close to zero, which essentially indicates a reductive or oxygen-free environment. So I got interested in how do you produce an embryo that can handle this kind of variation, and thinking about the types of things that can alter developmental outcomes. So we think about all of the genes that are important in regulating development and, for patterning various structures and physiologies. But there's also, in a lot of ecological interactions, random chance. Like in rats, it depends on who your litter mates are in the uterus. If you're a female that's surrounded by two males, you're going to have different behavioral properties as an adult. And that's just by random chance that you happen to get situated between two male siblings. Environmental variation, physiological variation. Then there's the whole behavioral aspect of it, where your parents can choose to put you in different places and give you a different experience as an embryo. So as a physiologist and sort of biochemical physiology type person, I got super interested in thinking about the sort of knife's edge that embryos have to navigate, where they have to buffer things enough that they can faithfully reproduce the developmental program, but they also need to be flexible enough to pay attention to the environment or these interactions and use them adaptively to induce new phenotypes or alternate phenotypes. And so that's what got me started down the killifish route. So annual killifishes, I call them extremotolerant vertebrates. As extremophiles go, vertebrates are wimps. I admit that right now. I work in a department with a bunch of people that work on thermophiles and things like that. So yes, they're not thermophiles. But as vertebrates go, these things can handle 3 to 4 months without oxygen, 2 years without access to liquid water, pHs all the way down to 2, all the way up to 10 or 11. And salinities anywhere from distilled water all the way up to 20 times seawater. So they can basically handle anything you throw at them. In fact, I left some embryos in a petri dish once, and they dried up, and there were embryos alive encased in sodium chloride crystals on the edges of the petri dish. So these things are tough. They are found they're sort of a Gondwanaland origin. So they're found on both Africa and South America. And there are theoretically multiple origins of annualism, this complex life history that I'm going to show you in a minute, on both continents. So this isn't something where there was a common ancestor that just got separated. The idea, at least based on phylogenetic analysis now, is that this has evolved multiple times, so probably convergently. They're also pretty. So they're some of the prettiest freshwater fish out there. This is probably important to them since you'll see pictures of their ponds. They're the color of coffee with milk in it. So you really can't see. And I'm assuming that they use these colors. This is not my area of research, but there's a lot of people that do sexual selection work in these fish as well. You don't see them in pet stores because they're called annual fish because they live less than a year. In fact, some of them only live about 6 weeks. They can complete their whole adult life cycle in 6 weeks and then they die. And so most people don't like to pay a bunch for a fish and then have it die 6 months later. It's hard to explain to your kids. I couldn't explain it to my son when I brought a killifish home, so I just kept bringing a new one home. And it was his name was Gil, and I finally admitted this to my son not too very long ago, and he's forgiven me for it now. But yeah, it was yeah, Gil lived for like 10 years. And then after I got tired of doing that, I just would bring Dylan, my son, into the lab and say, oh, there's Gil right there. He's like that fish. So anyway, yeah, I have some Eli, don't tell him that. Anyway, so these fish that I study, Austrofunius limnaeus, are found in Venezuela. So the state of Zulia, which is in sort of the northern western part of Venezuela here. Here's a typical pond, and they live in coastal deserts or savannas. So here's typical dry or rainy season habitat. When I say rainy season, it means there's probably 2 or 3 episodic rains that flood everything with a foot or more of water. And then all that washes out to the ocean. This is like 2 miles, 3 miles from the ocean. And then there's these ponds that are left, and that's where you find the fish. Dry season habitat. I think you'll agree with me that this is not a place where you would expect to find fish, right? So they survive in any given pond, locally at least, by embryos that are encased in the mud and can survive for years without access to water. All the adult fish die and are completely removed from the population when the pond dries. So how do they do this? They evolved a life history that's very similar to a lot of insects and invertebrates, but very rare amongst the vertebrates, okay? So they produce embryos that stop developing, they turn their metabolism off, they go dormant, and that's called diapause. So what's really interesting about these fish is that they produce their embryos can stop at 3 different stages of development at diapause. So there's diapause 1, which is early in development, and this is like think of a totipotent or pluripotent stem cell population of cells that are just kind of floating around on the embryo. So they can stop there. They can stop at diapause 2. This is like the toughest diapause. In this stage, they can survive without oxygen for 100, 150 days and without water for years. And then if they break this diapause and move on, they can stop at diapause 3, which is essentially a fully formed fish embryo. It's waiting for whatever cue it is to hatch. For these guys, it seems to be hypoxia, which makes sense in the terms of their habitat. The other thing that's cool about this is that they also can produce embryos that skip this and go directly to diapause 3 or hatching. That's called the escape trajectory. I didn't name it that. Everybody gets confused by that. It's because they escape diapause. Okay, so you can, and any one individual in the lab at least, will produce some mixture of embryos that use the escape trajectory and some that use the diapause trajectory. Okay, and then just to make the point about them being tolerant of things, during the rainy season, the embryos are probably hypoxic or anoxic almost all of the time because they're in the pond sediments. And during the dry season, of course, you still have anoxia because the clay is so thick. They live in a special kind of clay that basically shrink wraps around the embryo. And so it doesn't really leave a lot of space for, there's no interstitial space. The particles in the soil are really small. Okay, so this occurrence of 3 different places where they can stop developing is really unique amongst animals in general, because most organisms, even if you are going to employ dormancy to escape bad conditions, you have one life stage where you've invested most of that. So even if you look at insects, it's a larvae or it's an egg or it's the adult. It's rarely all 3. And for these guys, they have these 3 different diapauses. And so that led people to question, why 3, right? Why not 1? This one here is the toughest. It could theoretically get them through the whole dry season. So why not just have 1? And I apologize, when we put this on the thing here, this got a little fuzzy. But this guy back in 1972, John Worms, who's the guy that sort of originally described annual killifish and the occurrence of diapause, posited that this was essentially bed hedging on steroids, that you're basically he called it the multiplier effect. And by having 3 different places where the embryo can pause its development, you could create a huge amount of variation in developmental time. And therefore, at any one time, if you went to the pond, there would be some embryos that were ready to hatch or that were ready for whatever conditions were thrown at them, right? So that was the prevailing theory for a long time. And only recently have we found that it might be complete BS, but we'll see. Anyway, so this is the multiplier effect. And if you think about this, if you just had one diapause, you really can't increase your diversity that much. That would be one of these arms over here. But with 3 of them, you actually have 8 different potential outcomes. And then on top of that, even more diversity, because different embryos spend different times in diapause. Okay, so I got interested in this. Most of my work is done in the lab. I went down in 1995 to Venezuela, collected these fish, brought them back. We cultivate them in the lab ever since. And we started to do some studies to figure out, the real question is, what is it that controls? What are the molecular mechanisms that control entrance into diapause? So we started to do some studies, and we know that some females, so background information is that some females can produce embryos that won't go into diapause. Those are the escape embryos, right? And so we started tracking these in the lab. And what you can see is over time, this spawning number is like age, basically. So this is the first time they spawn, and this would be the 70th time. We spawn them twice a week on a regular basis. And this percent bypassing diapause 2 would be the same as an escape embryo. And you can see that early females produce a higher proportion of escape embryos. This is as a population. So this was an F1 generation, probably 42 females averaged together here. And then the interesting thing about these is that this F1 population here that's the offspring of a single female that was produced nothing but escape embryos. And we thought, oh, we had a spontaneous mutation. It's going to be great. We'll have a line that's completely doesn't go into diapause. Sure enough, her offspring recapitulated the whole realm of possibilities for going into diapause. And we tried this. This is the F2 generation. You can see we got disgusted when we started to see it again, so we stopped. Because for this fish, it took a long time. This is like 2 years. So take-home message there is probably some epigenetics going on in the system. All right. So we know that young females produce embryos that don't go into diapause more often than they do diapause embryos. We also know that no matter what the female produces, so we can take an egg from out here, let's say it should be going into diapause, and our normal incubation temperature is 25 degrees. We bumped up the temperature only by 5 degrees, 100% of those embryos will escape diapause. So there's another route, there's the direct escape route, and then there's another route that even though the embryo has been programmed to go into diapause, the environment of the embryo can override that. And we found out that it's mostly temperature. So we did some exposures over here where we did warm temperatures in the dark and warm temperatures in the light. You can see the control embryos down here. About 10 to 20% of the embryos at 25 degrees will escape diapause. But most of the effect is due to temperature, 33 degrees. And we gave them, this is just a 24 hours of 33 degrees and then back to 25 degrees. And that is enough, especially during this particular age class or this window of development, to cause most of the embryos to escape diapause, okay? And then if you put some light on top of that, it's even better. So you can get 100% escape. This is how we do it in the lab now. We just put them in an incubator at 30 degrees with the light on for 24 hours, pull them out, 90% of the embryos will have broken diapause and be on their way. Okay. So with this in mind, we decided to do an RNA-seq experiment. And I think based on this, I'm going to be missing a slide, but that's okay. And what we did is we took embryos and we incubated them in common garden at 25 degrees up until 100% of Pibili, which is essentially right before they're going to form an embryonic axis. And then we swapped them from 25 degrees either into 20 degrees or 30 degrees. And then this first point here is 24 hours after transfer. And then each of these time points is it takes about 24 days at 20 degrees to do this, and in about 6 days at 30 degrees to do this. But we would match them up based on their morphology and their stage, okay? So we're matching embryos that look exactly the same. We cannot tell them apart morphologically at all, looking for the molecular signals that are changing underneath to see what might be controlling whether or not they go into diapause or they escape diapause. And we're especially interested in this critical window. So I'm not going to show the experiments, but we did some temperature transfer experiments. And we found that if you put them in 30 degrees, by the time that they get to 16 somites, the decision to go on one trajectory or the other is set. You can't reverse it. So we know that by 16 to 20 pairs of somites here, the decision is made. Whatever happened, happened. Okay. I apologize because I put a slide in here that talked a little bit about the way that we did these methods, but it's not there, but that's okay. So we used regular RNA-seq, Illumina sequencing. We have a draft genome that we did. We assembled, I don't know, 10 years ago now, I think. And what we did was map the short reads, Illumina reads back to the genome to get differential gene expression analysis, right? So it's pretty simple, typical RNA-seq experiment. And then we used a network analysis called weighted gene co-expression network analysis. And what this does, think of a giant correlation matrix, and it takes every gene and correlates it to the expression of every other gene and lumps them into groups by their expression patterns. So a module in this, whoever made up this program liked colors. So the modules are all named after colors. You can't change that. I tried. I wanted it to be named after fish or something, but no, they're just these colors. So each of these colored modules is a group of genes that have a similar expression pattern. And in this heat map that you can see right here, the red is a high correlation coefficient in a positive, so it's highly correlated with this stage of development, that dispersed cell phase, that earliest developmental stage, only 24 hours after we transfer them from 25 to either 20 or 30. And then this is solid neural keel, so the embryonic axis is formed, and this is sort of early semitogenesis. They're just adding somites, creating a heart, things like that. What you can see is it was pretty easy based on this to categorize these modules based on their developmental stage. They were, as you might expect in development, the gene expression patterns are related to the developmental stage they're in. And this is the way that most people had used this technique, but we wanted to use it in order to compare what was going on in the 20-degree diapause bound embryos with the 30-degree non-diapause bound embryos. So the other thing that WGCNA can do is allow you to create a network. So we created the network based on the diapause trajectory. And this is just a heat map. Yellow means the gene was highly expressed. Blue means lowly expressed. And this particular one is this light yellow, the very first module in our analysis up here that's highly correlated with this first early stage, long before they're going to go into diapause. And so this is what that network looks like if you map it in sort of, I can't remember which particular mapping program we used, but basically the edges show interactions between genes or connections between genes. And the more close the nodes are to the center, the more central they are to the gene expression program, or at least predicted by their correlation coefficients. So interestingly enough, we came up with this DAF36-like protein or transcript for a DAF36-like protein that was pretty central to the diapause network. And when we look at this network, it's got an asterisk by it because this pattern was not conserved in the embryos that were raised at 30 degrees. So that tells us that this particular module is different between embryos that are going to go to diapause and those that aren't. All of the other modules are, and I say this is boring because I was looking for diapause specific things, but those are the things of development. Those are the things we expect to change because the embryo is actively developing, right? So they're important, but they're not important in terms of finding the genes that control diapause. So we ignored all of those. It's a whole nother data set that could be mined through because we didn't mine for that stuff. And anyway, when you look at the escape trajectory here, you can see that, you know, there's a high expression in the earliest developmental stage here. There's this group of genes up here that are more highly expressed than in diapause. And you can see that the shape of the network is really different. And that DAF36-like protein or transcript is now on the periphery of the network rather than the center. So this piqued my interest because DAF actually stands for Dauer formation. And Dauer is diapause, essentially, in nematode worms. So I'm like, well, that's kind of cool that we found a dormancy-related gene first, right off the bat, first module we looked at. And so let's look into that and see what it does in, whoa, there we go, come on. There we go. What it does in C. elegans. So for those of you unfamiliar with C. elegans, awesome little worm, probably one of the most powerful developmental models that we have. And if they have lots of food and they're happy and they're not crowded, they will just continue, they'll make these compounds called dafrachronic acids, which are like sort of a positive pheromone for, hey, everything's great, let's eat and reproduce. If you have crowding, starvation, high temperature, things that would normally induce diapause or dormancy and other things, they stop making dafrachronic acids, and that induces the Dauer larvae to form. Dauer dormancy is very similar to diapause in killifish, and it's controlled by a nuclear receptor similar to the estrogen receptor, glucocorticoid receptor, any of the nuclear receptors that bind to a ligand. And when they're bound to a ligand, some chemical, they go into the nucleus and they wreak havoc with gene expression, right? So DAF12 is the nuclear receptor that controls entrance into Dauer. DAF36, the one that we found, is the first step in the enzymatic pathway to make dafrachronic acids that activate this DAF12. And I won't go into the details of this because it's a whole talk in and of itself. But I will just say overall, the expression patterns that you see between these DAF or Dauer specific genes are consistent with the same types of expression pattern in a diapausing embryo compared to an escape embryo. The important point from this slide is that the vertebrate homologue for DAF12 is the vitamin D receptor, okay? Which was a huge surprise to me because vitamin D, everybody knows about this, you got to take it and take calcium so that you have strong bones, right? Which, by the way, is probably BS, but we could talk about that in another time. So we got super interested in vitamin D synthesis and signaling in these fish to figure out what's going on. So just a quick background on vitamin D. Again, you think of it as important for bone health, and what it really does is regulate blood calcium and calcium absorption in your gut. But vitamin D has been known for a long time to regulate lots of different things in the body, especially in terms of the immune system. It's important for heart development, brain development, but it's gotten very little billing. Like there's not a lot of work done on the effects of how it does this work. So we know that it is actually a very potent hormone. It's not a vitamin at all, right? Remember, vitamins are things that you can't produce in your body and you have to get from your diet. It turns out we can get it from our diet, that's fine, but we also produce it in our skin, right? So the way that humans deal with this, and I will say probably 80 to 90% of what we know about vitamin D is from human and mammal studies. Very little has been done in non-human or even non-mammalian animals. But it starts with cholesterol. The first step is 7-dehydrocholesterol. Okay, this is what that DAF36 makes, is 7-dehydrocholesterol. This sits in your membranes. You go outside, get a little suntan, maybe sun's out, guns out, UVB hits your arm, you get some pre-vitamin D, and if it's warm enough, it will actually isomerize into vitamin D3, cholecalciferol, okay? So this is the stuff, if you take a supplement, that's probably what you're eating. It's able to interact with the vitamin D receptor, but it's not very potent. Probably 2 or 3 orders of magnitude less active than the potent form, this 1-alpha 25-hydroxy vitamin D3. So there are 2 enzymes. In humans, there's one in the liver, a 25-hydroxylase that adds an OH to the 25-carbon. This is the stuff that if you go and get a test at the doctor and they say, oh, your vitamin D is low, this is what they're measuring. This is the circulating form in humans. In fish, the circulating form is actually 125. So in humans, the kidney would then take this 25-hydroxylase and add another hydroxy to the one carbon, and you get the active calcitriol. This is oversimplified. In humans, at least, the only tissue that has all of the synthetic machinery that you need to make vitamin D is your skin. Every other organ has only one or some subset of these enzymes. Okay, so that's sort of the background. And now you see where we're thinking, why didn't we think when we saw that light and heat were affecting development in these fish, why didn't we think of vitamin D before? Because, well, I don't know, vitamin D is supposed to regulate calcium. So we did the very simple experiment of soaking embryos in various forms of vitamin D metabolites. So if you just look at the top here, cholesterol, 7-dehydrocholesterol, vitamin D3, 25-hydroxy, 125-dihydroxy. And these are embryos incubated at 20 degrees and 25 degrees. Now, at 20 degrees, 100% of them should go into diapause, okay? If we don't mess with them and give them any vitamin D, 100% of them go into diapause. That's the blue. The orange are embryos that are escaping. So you can see that as we move down towards the more potent forms of the hormone, you get more and more orange, right? So we can definitely induce escape trajectory by just giving them vitamin D. And we also did some experiments where we blocked vitamin D synthesis, and you can do the opposite. So you can put these embryos at 30 degrees where they should escape diapause. If you block vitamin D synthesis, then they won't go into they'll go into diapause. Sorry. So so these experiments were great. Follow that up. The other thing I want to point out now is over here is the gene expression patterns of the enzymes that actually make these different forms of vitamin D. And note that at 20 degrees, see the blue down here? These enzymes aren't turned on at all. They're never expressed during early development. But if you plop the embryo into 30 degrees, you can see a nice peak of transcript being made for vitamin D synthetic enzymes, okay? So now we've got a situation where we can regulate the developmental trajectory of an embryo based on its temperature, and it's not just its temperature, but whether or not it's synthesizing active vitamin D. So in a situation where you take an embryo, no matter its origin, and put it at 30 degrees, it'll make vitamin D, and that will bind to the vitamin D receptor, that binds to the DNA and activates the program for active development. In the absence of vitamin D, this is another cool thing about the vitamin D receptor. A lot of nuclear receptors don't bind DNA if they're not bound to ligand, but vitamin D receptors do. So even without ligand, it'll bind to DNA, but it binds to different spots because it works with different partners and co-repressors. So that's our model. Little vodka in the afternoon is always good. Okay. There we go. You guys are still awake. That's good. Okay. So that's the first story. We get vitamin D signaling definitely controls diapause. But what about the situation where, if you look over on this part, what about these females that can change the distribution of eggs that they're producing? What's the female imparting into that egg that might regulate whether it goes into diapause or not? And I will say these experiments are a pain to do because I can't predict on any given day what types of embryos a female will produce. So the way we do these is we collect embryos, we freeze away 90% of them, we keep 20 or 30 of them out, and we monitor those. And then based on whether or not they go into diapause or not, we will sample those embryos that we first froze away. And so we only take embryos where 100% of them were either diapause or escape. It's a little bit of a stretch, but it's the best that we can do, given that we can't predict what a female will actually produce. So we got to wondering, well, what about a female? Like, if it's nice and warm and you're, eating a lot of stuff, maybe the female has a lot more vitamin D and maybe she's packaging more vitamin D into the eggs. So we actually measured vitamin D levels in females. And if you look in the liver and the ovary, you can see that embryos, so these females, we collected their eggs and then we sacrificed the female. And then once we saw that the eggs were going to be in one trajectory or the other, we took the actual female that made either 100% escape or 100% diapausing eggs. So from the females that produced 100% escape embryos, their ovaries and their liver are packed full of vitamin D, about double the amount that's in the embryos or the females that didn't produce those. And that doesn't really track across to the more hydroxylated forms, and there's probably reasons for that. Vitamin D is very potent, and so usually where it is actually active, it's deactivated almost immediately. So there's another enzyme, a 24-hydroxylase, that inactivates it almost immediately. And it's usually the level, sort of the enzymatic levels of the deactivating and the activating enzyme that determine whether or not vitamin D will be active in that tissue. So anyway, so it looks like they could. They're at least producing more vitamin D or accumulating more vitamin D from their diet. We're not sure what the origin of it is at this point. In those females. And if you look at their eggs, sure enough, the eggs are packaged with more vitamin D than an embryo that's not going to, that goes into diapause. We didn't see any of the hydroxylated forms in the eggs at all. So whatever they're packaging is probably this vitamin D, if this is the mechanism, which we think it is. And this is, remember, in just a one-cell stage embryo. So this is yolk. The mom has definitely packaged the stuff in there, either intentionally or not. It could be just based on a lot of hormones go into eggs based on blood levels in the females. Some of them are actively accumulated, so we don't know any mechanisms yet. But this makes complete sense when you look at the. Are you going to go? There we go. When you look at this, so if the female wants to try to produce escape embryos, that's great. She could put a bunch of vitamin D into the eggs, but nothing's going to happen unless you get at least 30 degrees so that you can turn on the enzymes that are going to take that vitamin D that was packaged and activate it. So this is a great way to make sure that the mom has some say in what's possible for the embryo, and that the environment then has some input on whether or not it's appropriate for what mom's decision was to be carried out. Or maybe it's not. Maybe it's too cold. It's not going to be good. And so it's like sort of a maternal offspring conflict, and the environment wins. I'll just say that. Okay. There we go. So now we've gotten this where we're looking at younger females are probably these unknown maternal factors. At least one of them is very likely vitamin D. And when they package that into the egg, it's much easier to get an escape embryo, except if you get into some situation where the environment isn't appropriate, and then it'll go into diapause. There are probably things that the older females are packaging in, but we haven't looked at that yet. Okay, so that's that part of it. But what about now in the case where you've got elevated temperature? So go back now to thinking about any of these embryos. I mean, we can really reproduce this pattern no matter what. And so what is it that's so important about 30 degrees that could be causing these changes in the expression of these synthetic enzymes and everything? So I had a student that was really interested in small non-coding RNAs and their ability to regulate gene expression. So we did some small non-coding RNA expression analyses. The most important thing here is to see that most of the transcripts that we're looking at here were at the 22 nucleotide length, which is classic microRNA, for those of you that study this. And then the other important thing is to look down here at this panel. So you can see, like in this early dispersed cell stage embryo, 80% of the sequences from those embryos, those microRNAs that are in those embryos, are one microRNA, miR430. So that is a massive abundance of a small RNA. Okay, it's very odd to have that much of any one thing, and that goes down over time. And these other ones for miR10 build up. miR10, this makes sense developmentally because miR10 regulates the Hox genes. So we expected to see those come on and regulate the Hox genes, especially during seminogenesis here. But it was really this miR430. We're like, what is this miR430 doing? Why do we have so much of it in the embryos? So if you look, here's another heat map with yellow being high expression, blue being low expression. So you can see in the escape embryos that there's a bunch, like there's this lots of sustained high expression of these miR430 microRNAs. And there's a bunch of them. It gets really complex. There's like 4 or 5 different subvariants, and they're found in these multi-gene clusters with like 15 or 20 sequences in each one. But what's important is we would expect more miR430 to show up because miR430 is important for clearing maternal transcripts after early development. So we expected to see it here early on, we didn't expect to see it maintain. And if you look over here, these are just line plots showing the expression patterns. So almost across the board, the MIR430s are much higher. Look at this, 600,000. So we got probably 2 million reads total in these libraries that were mapped to the genome and everything. And of those, almost 600,000, so a huge proportion, are just these few sequences for MIR430. So this became an important focus for us. And we found it super cool that MIR430's job is to clear maternal transcripts. So almost all vertebrates go through this. We have what's called a maternal to zygotic transition. So the early part of development is controlled 100% by the maternal genome. She packages a whole bunch of proteins, mRNAs, and other things into the egg. That way you don't have to activate gene expression. You can do quick cell cycles, right? And just take one giant cell and make a bunch of smaller cells. So during that time, the zygotic genome is silent. When it's time for the zygotic genome to activate, MIR430 is expressed, and it goes, there are seed sequences in the 3' untranslated regions of the messages for these things. It goes and finds those, marks them, and they get degraded. So that you can't have a conflict between what the mom's programming was and what the embryo needs to do now, right? So you got to clear out mom's signals so that you don't have confusion basically in the embryo. So we came up with the idea that perhaps, and this is, we have not actually shown this yet, so this is a hypothesis, that there are maternal factors that are basically keeping the expression. There are suppressors that are keeping those vitamin D synthetic genes off. And at the maternal zygotic transition, those transcripts are degraded, that there's a de-repression. You get to synthesize those proteins. You can make active vitamin D. You can develop. So we're in the midst right now of working out that hypothesis. So stay tuned on that. Okay, and then just to show you how promising we think this is. So these are all maternally, we did an experiment where we looked at what mRNAs were actually packaged into the eggs. And the ones that are in pink all have MIR430 seed sequences on them. And all of these, every one of these genes that's in here is known to be important in regulating dormancy in other systems. So it's very high likelihood that we're going to find some interesting stuff in here, but that's yet to be seen. Okay, I'm going to switch gears now and talk about how all of this relates to natural conditions, because this was all in the lab, right? So right now at this point, we know that vitamin D, so light and heat, can affect developmental outcome. That seems like a really decent way to regulate a bet hedging strategy. You can produce some embryos that go into diapause, some that don't. But does that actually translate to the field? So there's me looking at my little field microscope. I think that cracks me up. But so we went out to the field. These are pictures from Uruguay. These are pictures from Mozambique, Africa. And we dug fish embryos out of the soil and spent hours sifting them through basically like gold planing for eggs through a little mesh. So you got to kind of love your work to do this. And it's also remind you that most of these ponds are found in fields that are full of cows. So it's essentially liquefied cow poop that you're sifting through, and you can really tell the difference between if it's a natural pond with cows or maybe one that's had an elephant in it or something like that. Everything's got its own je ne sais quoi, I guess. So, and if you look, so this is just some mud that we dug up, and it doesn't show up very well here, but that's a killifish egg right there. So the really question was, what is this thing actually experiencing in the wild, and does it have anything that approximates what's happening in the lab, right? So this is what that work looks like. This is a pond that was just trampled by, I think it was elephants and wildebeests or whatever, and then we come in there and trample it some more. So if this was, this right here was in sort of southern Mozambique, so right down in this area. And I apologize again, I don't know what happened to the resolution of this there, but it's not important because all you need to look at is the white bar over here, end of the rainy season. So this is, the ponds are still there. There's some water in them. we can find fish so that we know fish were in that pond that year. So we feel good about that. We dug up the mud, we took it back and we sifted through it. And early in the year, almost 100% of the eggs, both in Mozambique and also up in Tanzania, I didn't do that sampling regime there, but we're in diapause 1. So in the lab, my fish never go into diapause 1. Most annual killifish don't produce diapause 1 embryos in the lab. So this is one big hole in our understanding already. It's probably induced by hypoxia in the soil, but also there are studies that if you just leave eggs in a tank with the adults, something that the adults produce tells them to not develop. So it's kind of a mechanism. Well, these things will eat anything, right? So they don't care if you're a larvae that's even their own progeny, your lunch if you come up. So it might be useful for them to stay in the soil. But anyway, we were surprised by this because this is not just in the areas where the soil still has water on it, but on the edges too, where there's some mud that's starting to be exposed. If you come back later in the peak of the dry season, 100% almost. There's a few odd ones. These 2 ponds right here still had some moisture in the bottom. And so we think that's why there's still some diapause 1s, but most of the embryos are in diapause 2. That wasn't surprising to us. That's the most tolerant form. That's what we expected to see there. And then when you come back at the end of the dry season, so we come back, let it rain a few times, don't let the pond fill up all the way because then the things will hatch and you can't really sample them very well. But just let them get moist enough that some of the water can move back in, and you start sampling them and you'll find most of them in diapause 2, but you'll now start to see some that are have broken diapause 2 and are headed towards diapause 3 or in diapause 3. That's this color here. Interestingly, a lot of them are dead. The mortality of these embryos must be extremely high, right? If you're in the top, I don't know, 3 or 4 centimeters of the soil, you're not going to make it. The ones that make it are buried somewhere like 10 centimeters down. And so there's a whole bunch. I'm not a real ecologist. I just play one every now and then when somebody says, do you want to go to Africa? But they think that there must be some mutualism between large ungulates and the killifish. Like the ungulates come and, I mean, I could imagine that an elephant can push you quite a ways down into a pond if it steps into it. And a lot of the places where we sampled, you're like elephant prints and you're sampling it. So they think that they actually have to get pushed down into the soil. I don't know about that, but I will say that when you look at them in the shallow parts of the soil, they don't seem to survive very well. So we did the same thing. We went to Uruguay. I've got colleagues there, much safer than Venezuela. So I go there now. And we looked at the same thing. We see exactly the same pattern. You go early in February, it's the flood season there. Almost all of the embryos are in diapause 1. Note that in South America, it's a little bit more messy than it was in Africa. This is a lot less of a extreme environment. The moisture is a little higher generally in year round. So maybe there's a little bit of chance for bet hedging, but definitely the environment trumps almost all bet hedging that could be happening, at least early in development. So the pond is flooded, you're in diapause 1. If you go there in February 2020, a year later, the ponds happen to be dry that year. And when we sampled them, and you see that you get a lot of the embryos in either diapause 2, or that's this color in the middle here. Some of them were post-diapause and only a few in diapause 1. So South America is a little interesting. Maybe there is some chance here that you could do some bet hedging because there's a little bit of play in what you might find in a pond. But in areas that are super extreme, where you might think bet hedging would be the most important, at least theoretically, we don't see it at all because it's completely controlled by the environment. Okay, so after I found that out, I thought, well, crap, like we've been doing this work in the lab for like 20 years and maybe it doesn't really apply. So of course, I won't want to give up on all the work I've done. So I got to come up with a new hypothesis, right? So the new hypothesis is that, and also I bet somebody out there is asking themselves, how could vitamin D synthesis, which requires light, be important to an embryo that's buried in the mud, right? That's something that I've avoided and luckily somehow been able to talk my way around in all reviews of papers. But there's another theory that we have that I actually think might actually be better. So there's a pond in Mozambique. just to remind me that we're going to talk now about environmental or ecological ways that vitamin D could work into this story. So remember, vitamin D in humans at least requires sunlight, right? And there are a few organisms out there that it's still controversial whether or not they make vitamin D with or without sunlight. There's people that argue they get it from their diet. There's other people that argue they don't. I think they're going to find that there are mechanisms for making vitamin D without UV light. Okay, but for the most part, for what we know about like terrestrial organisms anyway, plants and animals make provitamin D3, this 7-dehydrocholesterol. It's in their membranes. It gets hit by sunlight. It makes vitamin D. Fungi do the same thing. They don't have cholesterol, they have ergosterol. That's where we get vitamin D2 if you're vegan and don't want to have the animal products. Same thing happens. It's basically a photodegradation product, right? So all your membranes have this. You get hit by light. You could think of it as, hey, this is a synthetic mechanism for making a hormone, or you could think of it as this is showing that there's damage. You're actually being damaged by light. So we thought, well, what would that mean in a ephemeral pond environment like this? There we go. And then just to, how did it do that? That was a graph. That's hilarious. It's from the same paper though. That's weird. Okay, anyway, that's good. I don't need graphs to show you things. A little bit about vitamin D. So it's ancient. I mean, everything down to fungi and probably even bacteria. There's some bacteria that make vitamin D. There's been some question about how useful it is. There was a movement in the sort of 70s and 80s to change its name to Soltriol to get people away from thinking of it as a vitamin and think of it more as a hormone. I don't think that ever caught on. But I like it. It sounds cool. The point of this slide was to say, like, if you're thinking about an organism that's in a seasonal environment, especially in the temperate zone, what you would have seen here is a graph of the quality of light over the change in that quality of light over the seasons. And what you would see is that if you go over here on the visible spectrum and imagine going over here to the UV spectrum, is that really for any unit of light that hits the Earth, everything down to UVA is relatively flat, right? I mean, light is light, comes from the sun. But because of the way because the Earth's axis is tilted, the angle with which the sunlight hits us changes through the seasons. And it's really that incident angle that changes, and it either deflects the UVB or lets it through. So when you get down into the UVB, imagine a line going way up really high. The UVB, so if you were going to create a sensor that was like super sensitive and told you what time of year it was, you would want to sense UVB. That would be the best way to tell if it's winter, summer, whatever. It would be the largest change in between seasons. So just thinking about all of that as I was thinking about how this could work in the ecology of these ponds. So we decided to do something crazy. We dug up a bunch of soil and extracted vitamin D. Well, extracted anything that would go into acetonitrile. That's what this is right here. So we did these extractions. Actually, this pond right here is the local frog pond from my neighborhood. I tested this out when it was dry, and then after it got moistened. But we've since done this in the field in Africa. We haven't made it to South America yet. But you can actually find vitamin D extracted from the soil, and it's very different in dry and wet seasons. So if you look at just that transition between late dry season, so late summer here, and early wet season, so this was taken in late August, this was taken in early October. You can see in this little area here, here's our standard for vitamin D2. You can see a nice little peak of it showing up. It takes a lot of soil to get enough of this, so it's not a trivial task, but it's measurable. So we went out to Zambia, got some field sites in Zambia, Africa this last May, and collected those, and we're in the middle of analyzing those right now to see. We went to that spot because there's a very high gradient of moisture from the south to the north. And so we were able in one trip to sample ponds that had been dry for a long time and ones that had just been wet. And so get as high a diversity as we could, at least in the first shot. But unfortunately, I don't have those data ready yet because the lab at OHSU that runs these for us. So they have a nice mass spec protocol for this now that's highly sensitive, but it flooded. So we're waiting for them to repair the instruments, and then they'll run our samples for us. And you can imagine that when there are people that want to know their vitamin D status for health reasons, that soil samples get put on the back burner. So I don't know why. So interesting, like this maybe is not surprising, but vitamin D2, it was the major stuff that we were able to find in the soil here in North America. Vitamin D3 didn't show up at all. I suppose that makes sense. There's probably not a lot of plant matter in that area, but there's obviously a lot of fungus in any moist soil. So that's our working hypothesis at the time. And we're hoping to see, the idea would be that that vitamin D will actually soak down into the soil and activate the embryo, so it'll start developing once the soil gets moist. And then, a few weeks or a month later, when the pond actually becomes inundated, they're ready to hatch. Because if you go to these pools, the one thing that's really amazing is that we see them in diapause too up until the end of the dry season. But if you put water in that pond, they're up within 5 or 10 minutes. There's fish hatching out of the soil. I mean, they're amazing. So there's obviously fish in there ready to hatch at a moment's notice, late in the dry season, early in the rainy season. Okay, I'm just going to show you a couple of things that we're working on now that aren't about vitamin D. So this is what I did for my year off in between doing administrative work and back to the faculty. I got super interested in space biology, and I'm like, these killifish are so awesome. We can pack like 1 million of them in a 1 square cube box, and you can send them to Mars, right? Well, it turns out it's harder to convince NASA to give you money than just saying it's a great idea. So I had a chance to go and do some experiments at the NASA Space Radiation Laboratory. So if you think about what's limiting in space, it's water, oxygen, food, physical space, right? So water, oxygen, food, no problem for diaposing embryos. What's different in terms of and expensive to control? Of course, there's no gravity, ton of radiation, and there's no magnetic field. So I started to ask myself, well, what can we look at? So we're looking at radiation and magnetic fields right now. So just to convince you that these embryos are tough, they can handle genotoxic stressors that would shred your genome. Like they can live long-term in 1% hydrogen peroxide. They can live for a couple days in 3%, like just pour the stuff out of the bottle from the pharmacy. Gamma radiation, we put them in front of a cobalt source, up to 200 gray, they survive. Up to about 25 to 30 gray, they're completely normal down here. So to give you an idea, a 5 gray dose of this would kill you. So they're 5 times, 20 times better than us. UV radiation, we put them under a germicidal lamp for 45 minutes. Their DNA gets completely shredded. It's got pyrimidine dimers all over it. 24 hours later, not a dimer left. They're fine. They have this great photolyase DNA repair enzyme that I wished I had. That would be awesome. Okay, so we went to NASA. This is their space radiation laboratory. So they're hooked up to Brookhaven National Labs, which has got a particle accelerator. This is the actual accelerator thing here. It was super cool. I totally geeked out on this. Here's what their little radiation lab looks like. If you look through that window right there, that was the particle accelerator. And so the radioactive beams come through here and you put your sample right there. Yeah, this is what the accelerator looks like. It's just these magnets, and there's a whole bunch. They actually do physics on paper to figure out dosages and stuff. It's pretty crazy. Anyway, they can expose embryos there to simulated galactic cosmic radiation. That means lots of protons, because that's mostly from the sun, but iron, like radioactive iron, which is like, think of a little molecular bullet. When your cells get hit by that, it goes clean through them, leaves a scar all the way across and heads out the other side. There aren't very many of them, but if you're going to spend a year in space, you're going to get hit by radioactive iron eventually. So this is what it looks like. Here's my little tubes full of embryos. This is an actual picture. I don't know what it means, but they're like, oh, this is what the beam looks like. I'm like, oh, it's pretty colors, so I'll put it on there. And there's the embryos there. So we exposed them to 4.5 gray dose. This is 5 times what you would get if you went to Mars. 1 gray dose would make you super sick. Maybe you wouldn't survive it because your immune system would be dead. 100% survival, no abnormalities. Developing in diapause, doesn't matter. These fish are tough. So we're in the middle right now. We're waiting for the RNA-seq data to come back. I wish they were back now, but still waiting for that. And then the last thing I'll say, this was a crazy trip I took. I met these quantum biologists in this NASA chat group, and I'm like, okay, this is either going to be super fun or crazy, right? Because I don't even understand what they're saying half the time. But they have this chamber called a mu chamber. It completely erases the magnetic field of the Earth. Like there's a lot of studies out there of inducing higher magnetic fields, right? And anytime you go and get a medical, like an MRI or something, you're in a giant magnetic field. So think about a magnet size for an MRI is like 1 to 5 Tesla or something like that. The magnetic field of the Earth is 50 microtesla. So most people don't think that could have any effect. And they're probably right on average, right? Because so the thermal noise in biological systems is huge compared to that. So, but there are some quantum effects on certain proteins in your body that might affect that in the absence of a magnetic field. And they've actually shown that birds navigate through the magnetic field of the Earth using quantum effects on their eyes. Okay. So I thought, why not? I got some time. I got a little extra money left in my sabbatical thing. I'll go down to LA. We'll put some embryos in this mu chamber and we'll block out the magnetic field of the Earth. And sure enough, it completely messes with their ability to break diapause. So hypomagnetic here is basically zero magnetic field. You put them in that chamber and they stay in diapause way longer than you would think that they should. If you put them in there before they go into diapause, they will go into diapause. It's like putting them in a low temperature, even though they're at 25 degrees. So there's clearly an effect of a lack of magnetic field on their development. I don't know what it means yet, but I do know that it means that if we're going to send people out to space, it doesn't take very long to get outside of the Earth's magnetic field. We don't know whether or not we're going to be able to reproduce out in space, right? Elon Musk might be wrong. Go figure on that. So anyway, this is just kind of a fun thing that we're doing. I'm trying to get money from NASA right now for this. We've got a consortium of people that are working on sort of space biology to figure out what is actually possible. The nice thing about this is if it is a problem, it's fairly easy to fix. You can induce a magnetic field. You just need to know you need to induce it, right? It'll be that one thing that we don't think of that'll take us down, right? So we got to think of everything. All right. So with that, I'll be quiet. I'll thank this is my lab. They did most of this work. My collaborators from Czech Republic, from Uruguay, these are the physicists, and Afshin's the guy that's working with me on the NASA stuff. And I'll take any questions. Thanks. Oh, lab motto. Yeah, Virginia.
Every time I code, there was EGF beta.
Mm-hmm. Yep.
So my guess is that, and you mentioned maybe that might be another really important.
Yeah. I totally agree with you. And one of the other genes that's in that light yellow module is interleukin 1 beta. Yeah. So I totally agree with you. And I think the immune story on this is super interesting. So when we look at, we took some diapause embryos and we put them into anoxia. And one of the big signatures that comes up when they're stressed is it's all immune response, even though in theory, there isn't a developed immune system yet, right? In this stage, they might have these embryonic macrophages that crawl around and stuff, but there aren't differentiated immune cells at all. So I think, and we were, talking over beers that maybe, the immune signaling pathways are actually ancient developmental pathways that were kept in the immune system and lost in other things. I mean, I can come up with all sorts of crazy ideas, but yeah. Yep. Also important in regulating, there's a pathway that regulates DAOR that's TGF beta control. So thank you. Anybody else? I answered every question that you had. Wow, that's great. Yeah.
So you said you were trying to, or maybe thinking about looking at the difference between the eggs produced by older and younger females. And so I was just curious, how many reproductive events occur within a female's lifetime?
Yeah, so these fish, they reach sexual maturity super fast. I mean, they can get there as fast as you can feed them. If we feed them multiple times a day, we can get them to sexual maturity in like 2 weeks. And then they will spawn every day until they die. So we spawn them um, twice a week, and they normally live about, so that's what, 2, 4, 6, 8 times a month. And they normally live about a year. After about a year, even if they're still alive, their reproductive output goes down quite a bit and fertilization rates go down. So there's a lot of reproductive events. And what's interesting is that, like I said earlier, you can see almost any pattern of egg production. Like some females will produce escape embryos their whole life. Some of them will produce diapause embryos their whole life, and then everything in between. And you can't predict any given day. It probably has to do with what we're feeding them, although we feed them a pretty controlled diet. They won't eat pellet food. They're very picky. They like frozen bloodworms, which is probably the most expensive type of fish food you can imagine. I spend like thousands of dollars on frozen bloodworms every year. But twice a week, we feed them earthworms, like compost worms. So they're probably getting who knows what from that. But if we give them that, it ups their egg production a lot. So that would probably tell us that they're putting something in there. Maybe I need to do vitamin D analysis on the worms. Yeah, yeah.
So you want to throw away that hedging?
Yep, throw it away.
A lot of advantage of having multiple tactics and strategy of dealing with really environmentally heterogeneous environment. And so,
Yep.
Yeah, so then what fills that gap other than just having a lot of phenotypic elasticity?
Probably bet hedging. No, I mean, I don't know if it was the year that we were there and it was really, we're obviously going back to sample some more, if it was just super harsh that year or not. But I actually do think that the bet hedging happens. It just happens at the end, that like there's a very short window for bet hedging between when diapause 2 breaks and diapause 3. And basically, will you break diapause? I guess that's the bet hedging at the very end, right? Because you have to be in diapause 2 to make it through the dry season. If you're not, all the other ones will die in about a month or something like that. So it's really about, if you make it through all of that, then I guess there is a chance for bet hedging, because we know that not all of the embryos will go on to develop and hatch that season. There will be some that stay as a seed bank or an embryo bank. But it can't be the major way that they survive, but it might be super important only once every 10 years, and that's when you need it. And so it's there, right? Could be that on average years, it's really not important. But I have no other explanation that's, yeah. Mm-hmm.
Common equivalent to something like a zebrafish that doesn't, or do they have special mechanisms to get some place in development and all the other places?
Yeah. Maybe you were reviewer number 3 when we tried to publish this, because they asked us, why didn't you do this in zebrafish? And my answer back was, well, because zebrafish don't diapause, number 1. But number 2, it's like, so at first I was mad. I was like, oh man, I got to do it in zebrafish. But then I thought, well, this is great. We can do it in zebrafish. So we actually took zebrafish and inhibited vitamin D synthesis. We can't, I mean, they're already developing, so giving them more vitamin D isn't going to help. If you inhibit vitamin D synthesis in a zebrafish embryo, it stops at what's called the Prim5 stage, which is roughly equivalent to diapause 2. So it may be, and there was some arguments in the literature about, when to stop in development, what's the most resistant stage, why there, like why not stop before you have a heart and a brain and all these sensory organs starting to form? Why not stop earlier? I don't have a good answer to that question. I do know that it's right before the major sort of organogenesis endoderm development. And so maybe that's the sensitive part. But yeah, but that's where they stopped too. So that's actually what got us in PNAS, I think, is because the zebrafish stopped too. So we were like, yeah. Yeah.
So we all.
What?
Global climate change.
Mm-hmm.
Fish, is there any place like a great place to live? Like it's just like water there all the time and I don't have to dry out and I don't have to go like extreme temperature, whatever. are they sort of constrained to live this tough life?
Yeah.
They ever live in a really good fish habitat?
No, they don't. No, they've made the choice. So they've made a trade-off that they'll live. So there are some places on the the Guajira Peninsula between Colombia and Venezuela that have semi-permanent ponds. They might be there 3 or 4 years at a time. And what you'll see is that they'll be there for a couple of generations and then they just disappear. So they have to have at least periodic drying of their habitat. And in most places where you see this, it's, we call it an annual cycle, but it's probably happening multiple times within each season. So in Venezuela, when I was there, we came by a pond. It was almost dry. We collected some fairly large fish, and then we drove back by it. A few days later, it was completely dry. We came back a week later. It was full of water and it had little fish in it. So they're probably going through multiple inundations in a year, which maybe that gets into the bet hedging. I hadn't really thought about how that would work into it, like in that off chance that you dry too soon before everybody can reproduce. So yeah.
Flip it on it, you know, we're always, oh, fine, it's gonna wipe everything out, whatever. Like,
Yeah.
already used to be.
Yeah. Yeah. I mean, there's the question. Yeah. There's the question of whether or not they're already living on the edge of their ability. Right. And so, if we push them even a little bit farther, that might be a little bit too much, but I don't know. They're pretty tough. Like they definitely have evolved tolerance beyond what they have to endure on an average annual cycle. So. Yeah.
Thank Jason one more time.
Thank you.